Alright guys, this is going to be your test number 2 review, and so we're going to go through the review and I'm going to show some slides and hit on some high points that you need to study for this exam. So go ahead and make sure you have your review in front of you and let's get started. We're going to start with chapter 4, and when we're talking about chapter 4 guys, the first bullet says you need to know the function of the following structures. Okay, so the first structure that's listed on your review is the plasma membrane. So you need to know that it's a barrier and it regulates what goes in and out of the cell. Okay, so you need to make sure you know it's a barrier and it regulates what goes in and out. Also, the second one is you need to know the lysosome. Now the lysosome is a vesicle that contains hydrolytic enzymes. Now remember, these hydrolytic enzymes are going to help digest things inside the cell. The chloroplast. Now this is an energy related organelle. That's going to be important in 2 separate questions on this exam. They are related to energy, but they also perform photosynthesis. So that whole idea of converting solar energy into sugar, that happens here at the chloroplast. The nucleoid is the next one. It's the location of DNA in bacteria cells. So this is going to be important because it's found in bacteria cells, not in your animals and plants. Now cytosol. Now cytosol sounds like a little bit different, but this is just cytoplasm. It's the semi-fluid that bathes the organelles. So it's the fluid inside of the cell. We also call it cytoplasm. Okay, but in your book or on your test, it's also going to be looked at as cytosol. And then last, the mitochondria. Now remember, the mitochondria is an energy-related organelle as well, like the chloroplast, and it's going to generate ATP. It's also known as the powerhouse of the cell. Now guys, all of these things except the nucleoid are found on the lab assignment from lab 4, where you were supposed to color the cells, label them, and also tell me the general functions of the organelles. So that's going to help you with this as well, but these are some that are very specific that will be on your exam in the matching portion. Now, your next bullet says you need to know what cells have cell walls and what they're made of. So ignore the cellular junctions title for just a second. So what cells have cell walls? Well, first, plants do, and they have a cell wall made out of cellulose. Okay. Second is fungi. Fungi has a cell wall made out of chitin or chitin. Bacteria has a cell wall. It's made out of peptidoglycan. Okay, so that's going to be for bacteria. And then, of course, animals have no cell wall. All right, so a question may be something like, all of the following have cell walls except, well, you would pick animals. It might be more specific and say, this organism's cell wall is made out of peptidoglycan. Well, that would be bacteria. So you need to make sure you know which ones have them and what they are also made out of. All right, now your third bullet is know the types of cellular junctions in plants and animals. So now we can focus in on the title of this slide, which says cellular junctions. First cellular junction we want to hit on is plasmodesmata. Now this is a cellular junction found in plants. So that's what you see here in this picture. Now guys, the reason that you know that this is for a plant is you can see here that it says cell wall. Plants have cell walls, animals don't. So you should be able to tell that this is a plasmodesmata because this is a plant cell. It has a cell wall. Okay. Now in animal cells there are 3 types. There are tight junctions, anchor junctions, and gap junctions. The tight junctions are the zipper-like ones. It's like they've been sewn together. Okay, so they're tight. Anchor junctions is where they're going to hold on to each other. So they're anchoring. And then the gap junctions are just channels or holes that allow the animal cells to communicate. Now these are very similar to plasmodesmata. However, these are found in animals, so there'll be no cell wall in the picture. So you can see these here. You can see the tight junction where it's looked like they're sewed together or zipped together. The anchor, you can still see there's space between them so fluid can get through, but they are holding together. And then of course the gap junctions. Okay, so these are your cellular junctions. Now the fourth bullet on this chapter 4 guys says know the 2 organelles responsible for energy transformation. We discussed this previously. That's your mitochondria and your chloroplasts. Okay, we're going to switch gears and we're going to move on down into the matching here for chapter 4. Your first bullet in the matching says be able to match organelles or structures with their functions, and this is for plants, animals, and prokaryotic cells. Well, here's your prokaryotic cell. So I'm going to tell you which ones you need to really pay close attention to. Okay, you need to make sure that you can find the cytoplasm in a prokaryotic cell. And I know this slide is a little blurry, but cytoplasm is the liquid. Also be able to identify the ribosomes. Ribosomes are the ones that look like the sprinkles inside of the cell. Also know that the nucleoid is where you find their DNA. The 2 ways that these cells can move is through flagella, which is the tail you see here, and then the pili are the smaller extensions that help them hold on. Now, of course, these guys have a plasma membrane, a cell wall, and a capsule, but these can be kind of hard to see in a picture for a test, and so it may be where they're all listed together, and you just pick it'll be where they're listed together, and you just pick that option, but it's hard to tell in the picture to separate them, so I'm not going to worry about that part for them. Okay, so this is the bacteria cell. For the animal and plant cell, guys, I cannot tell you which one you're going to have. There's an animal option and a plant option, and it's going to just shoot it out to you randomly. So you need to be able to tell the different types of organelles that are found in them. Also, use your chapter 4 diagrams, okay, because those are going to be really important, and the structures look very similar on the test. Okay, so make sure that you pay attention to the ones they share in common. Okay, you know where the nucleus is. The nucleolus, guys, remember, is on the inside of the nucleus. Okay, the Golgi complex looks like the stack of pancakes. The rough ER and the smooth ER, the difference is the rough ER has the ribosomes on them. Okay, so pick out those differences. Also on the test, I may not give you the structure name. Instead of nucleus, I may say this organelle is the control center, and it'll point to it. And so you'll need to also know its function. So study that chapter 4 lab assignment, because that's going to help you more than anything on this part of the test. So you may have a plant cell or you may have an animal cell. Okay, so pay close attention to that. Now skip down to the first bullet in your short answer. Your first bullet in your short answer is talking about the difference between prokaryotic cells and eukaryotic cells. Guys, you can use any of these in the question, okay, to answer it. I'm going to ask you for 2 differences. So you could even tell me the difference in their domains. Prokaryotic cells have archaea and bacteria. Eukaryotic cells has eukarya. You could tell me the difference in their DNA. Prokaryotic has a single loop. It's found in the nucleoid region. Eukaryotic cells have chromosomes, and they're found in the nucleus. You could talk about their organelles. Prokaryotes don't have any except ribosomes. Eukaryotes have a lot. You can talk about their size. Prokaryotic cells are smaller and simpler. Eukaryotic cells are larger and more complex. Also, most prokaryotic cells are unicellular. Most eukaryotic cells are multicellular. So when you look at this, you have plenty of differences here, and you need to pick out 2 of them to discuss on your exam. Now, the next bullet for your short answers, you're going to need to be able to do a comparison between plant and animal cells. Now, this can also be a multiple choice question, where it might say all of the following are structures that plant cells have that animal cells don't accept. Okay, and so it may have an exception question. Remember, plant cells have a cell wall, chloroplasts, and a large central vacuole. Those are 3 things a plant cell has that an animal cell does not. On the other hand, animal cells have lysosomes and centrioles. Plant cells do not. Okay, so those are your differences, so make sure that you can tell the difference. Because you also might get an animal or plant cell, and it'll ask you, is this an animal or plant? Well, if it has a cell wall, a chloroplast, or a large central vacuole, it's a plant cell. If it has centrioles or lysosomes, obviously it's an animal cell. So make sure you study this chart as well. Now, another short answer that you might get is it may ask you, what are the 3 tenets of the cell theory? Well, here's the 3 tenets. All living things are made of cells. Okay? Cell is the basic unit of life. Well, that makes sense. If all living things are made of it, of course they're going to have that basic unit. And then all cells come from pre-existing cells. So these are the 3 that you need to discuss in that short answer. So make sure that you know all 3 of these tenets of the cell theory. Okay, so in your short answers, these were the first 2 bullets. Now let's go back and look at Chapter 5. Chapter 5 says you need to know the function of the plasma membrane and the structures that make up the membranes. Okay, well, here it is. This is straight out of your notes, guys. Make sure that you can tell you can label a phospholipid. Remember that the plasma membrane is a bilayer of phospholipids. If it's this honeycomb structure, you're talking about the steroid. If it's a protein, it can be one of 2. It can be an integral protein, which means it goes through the whole membrane, okay, like you see here. If it's a peripheral protein, it's on the edge, because remember, peripheral vision is out here on the sides. Now, remember the difference between a glycolipid and a glycoprotein. Glycoprotein is where this sugar is attached to a protein. Glycolipid is like what you see in this picture, where it's attached to a lipid. Okay, so know this structure. This is going to be a matching section. Okay, it may also say all of the following are components of the plasma membrane except, okay, if they end up telling you that waxes are a part of it. Nope, it's not. There are 2 fats, but not waxes. Okay, so make sure that you pay attention to that. Also, make sure you know the difference between hydrophobic and hydrophilic. This will be a potential short answer here. Remember, hydrophobic molecules do not like water. That's like your fats. Hydrophilic do like water, and that's like the hydrophilic heads you see here in the picture. Okay. Also, the next one, this comes again straight out of your notes. This is the types of passive and active transport and their requirements. Okay. Remember, passive transport are your simple diffusion, your osmosis, and your facilitated diffusion. Okay. They all do not require energy. You can see the no all the way across the board here. The only one that needs a carrier protein is the facilitated diffusion. And these all move from high to low. Guys, pay close attention to these pictures down here at the bottom. There is going to be a matching section for these pictures. Okay? If it's water moving high to low, pick osmosis. If it's something moving high to low through a protein, it's facilitated diffusion. If there's no protein, it's simple diffusion. For active transport, guys, they do utilize ATP. Also, a protein's needed, and they go opposite. They move from low to high. So pay close attention to these pictures for your exam as you're studying these. Also, there's going to be a short answer that's going to have a picture of either exocytosis or endocytosis. And it's going to say, is the following exocytosis or endocytosis? If the molecules are exiting like you see here in this picture, please pick exocytosis. If they're entering, like you see here in these two pictures, this is endocytosis. Remember that endocytosis for phagocytosis is when they take in large molecules, so it's cell eating. Penocytosis, on the other hand, is where they're taking in smaller molecules along with liquid, and we call that cell drinking, penocytosis. But whether it's phagio or peno, it's still taking things inside, so it is a form of endocytosis. Now, the next particular bullet on your review in Chapter 5 says, what factors influence how molecules are transported? Well, first, the size of the molecule is very important. Remember, large molecules have a hard time. Also, the charge. If your membrane's a positive charge, other positive molecules are not going to be able to get in because they repel each other. Also, the type of the molecule, if it's fat-based or not. And then, of course, the concentration gradient. Remember, molecules like to spread out. So if there's already a high concentration, things are not going to be able to get in. Okay, so these are factors that influence it. Your next bullet says, know what happens to a cell placed in an isotonic, hypertonic, and hypotonic solution. Know the scientific terms. Again, guys, this comes straight out of your notes. Okay, now remember, in an isotonic solution, iso means that everything is equal. So the cells are happy, whether they're plants or animals, they're just staying normal. Remember, in a hypotonic solution, because the O, the cell's going to get bigger. Remember, in an animal cell, this is called hemolysis. In a plant cell, this is called turgor pressure. Okay? Hypertonic. Remember on the board I drew the E and it was all shriveled up and shrinking? Okay, so when we look at this, a hypertonic crenation is when an animal cell shrinks. Plasmolysis is when a plant cell shrinks. Please pay attention to these scientific terms. Okay? And like I said as well, this is also explaining iso, hypo, and hypertonic, which are terms used to determine the tonicity of a solution. The last thing on Chapter 5 in the multiple choice portion is you need to know the roles of integral proteins. Okay? They can be channels. That means they're going to act as like a bridge. They can be carriers. They're actually going to grab the substance and carry it over. They can also be used for cell recognition, recognizing whether it belongs to you or not. A receptor, where it's going to receive messages. They can be enzymes, or they can form those junctions we talked about earlier. Okay? So again, this is going to be a multiple choice question that might say all of the following except, or which of the following is an example of what an integral protein does. Okay? So you need to know these. All right? You need to be able to identify them. Now, this brings us to Chapter 6. Chapter 6 says, know the two laws of thermodynamics. Guys, the first law says that energy cannot be created nor destroyed. It can only be changed. So that's the first law. Now, the second law, guys, is super long. This is straight out of your notes. But the key thing, guys, is that it means that sometimes it's going to be lost as usable energy. Usable energy, okay, is going to be lost in the form of heat. So heat is the important term here. If you see heat in the definition, it's the second law. Also, you need to know the definition of entropy. Entropy is the amount of disorder in a system. So make sure that you write that down as well. Entropy is the amount of disorder in a system. Okay, so you have your two laws of thermodynamics and entropy here. Your next bullet says you need to know the structure of ATP. You had to draw this in your portfolio. Okay, so you need to know that it has adenosine, that's what the A stands for. The T is for tri, and the P is phosphate. Well, that means you have 3 phosphates, and you can see them here in the pictures represented with P. Also with this, guys, is you need to understand how endergonic and exergonic reactions are linked to ATP. First, endergonic reaction is when the chemical reaction requires an input of energy. This is when you go from ADP plus a phosphate, and the energy is stored in ATP. That's an endergonic reaction. Exergonic, though, is the opposite, where energy gets released. This is when ATP breaks open, releasing energy into ADP and a phosphate. So be able to know that endergonic is where we're going towards ATP and we're storing energy. Exergonic is when ATP is being used and it's releasing energy. Now, the next bullet says know what entropy is. We already discussed this, but also says know what feedback inhibition is, guys. Remember, feedback inhibition is the method in which cells control the amount of product being made. So in this example, A uses an enzyme called AAce, which makes B. B uses BAce to make C. C uses CAce to make D. And D uses DAce to make E. E is our final product. When we have enough of E, you'll see that it goes back and it shuts down AAce. So the reaction stops. Once we run out of E, however, E unbinds and the process starts over again. Make sure that you understand that this picture is a feedback inhibition. The next bullet says know what affects enzyme activity. Well, first we need to look at substrate concentration. Remember, the substrate is what the enzyme's working on. So in the lab, catalase worked on hydrogen peroxide. Second thing is temperature. So you need to look at how temperature affects it. pH, acid versus base, and then also cofactors versus coenzymes. Well, this brings us to our next bullet, though. What is a coenzyme? Well, guys, cofactors are inorganic ions like copper, zinc, or iron. Coenzymes, however, are organic or non-protein, and so examples would be like NAD, FAD, and CoA. Guys, pay close attention to NAD. It's going to say NAD is used a lot of times in enzymatic reactions as a blank. It's a coenzyme, okay? So coenzyme. Now, the last thing I want to talk to you about before I move on to degradation and synthesis is, guys, remember enzymes are normally named with ACE at the end, A-S-E at the end, so make sure that you understand that. Now, this is going to be one of your short answers. It's going to ask you whether this is showing you a picture if it's degradative or synthesis. Now, guys, if an enzyme breaks the molecule apart, so here in the yellow you see the substrate, we start as one molecule and it breaks it into 2 products, that's degradation. Okay, degradation is to break. Synthesis is where we start with 2 substrates and we make 1 product. That's synthesis, where it's being built up. Again, guys, this is a 50-50 shot. The picture's there. It asks you, is it this or this? And you tell me which one it is. Okay, so 50-50 shot on this question. Please don't leave it blank. So guys, this kind of goes through and gives you an idea about the review. Okay, I went step by step through it as we went through this whole review, so you may have to pause or maybe rewind different things to be able to get every part of this, but I hope that this helps you prepare for your exam. On Monday during class, however, if you have extra questions, maybe you thought I didn't cover something well enough or you have specific questions, please let me know and I'll be willing to answer those on Monday. Have a great weekend and study hard.